continue from the same topic, from the same question where we left in the last video. It is related to paper chromatography. The question in front of you, it's a past paper question. And what this question is telling you, a molecule contains 4 amino acid units. These amino acid units are linked in the same way as a protein. A sample of this molecule is hydrolyzed. I have told you in the previous video as well that hydrolysis is a chemical reaction in which a compound is broken into its components by adding water. The resulting colorless solution is spotted onto chromatography paper. The paper is placed into a suitable solvent. A diagram of the final chromatogram is shown. So there are many points on which we should have a discussion right now. The point is that it is colorless. So what is this colorless telling you? That the spots on the chromatography paper are not seen. We cannot see those spots because they are colorless. Then there is another point, suitable solvent. It means that the spots which we have placed here should be soluble in this solvent. So there are 2 types of the solvents. In the previous class, it has been mentioned that there are 2 types of the solvents. One of the solvent is water, whereas the other solvent is alcohol. Depending upon the solvent, we are going to select depending upon the spot, we are going to select these solvents. Okay. So sometimes examiner is saying this thing that the spot is insoluble in water. In that case, we have to select alcohol. So right now we are going to select this spot over here. We are going to place this spot over here with the help of a pipette or with the help of a dropper. So what is this? This is called start line, and this is the point till which the paper is immersed. This is the answer of this question. I am telling it to you, but I will explain it further here. So just why the initial line is drawn in the pencil, again the same answer. As ink is soluble, it will affect the results, or it will spread on the paper. Draw a line on the diagram of the chromatogram to show the depth of the solvent into which the paper is placed. This is the line till which the paper will be placed into the chromatogram. Suggest how the chromatogram needs to be treated to make the spot visible. So we have to spray the locating agent. It is a reagent. Basically, you may say it's a chemical which will turn the colorless spots into colored spots. So locating agent is a specific reagent. It's a specific chemical which is going to react with the spots on the chromatography paper. Hence, it will turn the colorless spots into colored spots. Then, why does hydrolysis have on sample of the molecule? So what does hydrolysis have on the sample of the molecule? This is particularly not part of your current syllabus, but now let me inform it to you. I have told you earlier that hydrolysis is a process in which the protein will break up into its components. Let me inform you one more thing. Amino acids are a specific type of molecules which are attaching with each other to make protein, and protein is the building block of our body, for example. Okay, so when it is hydrolyzed, protein is hydrolyzed, it is turning itself into amino acids. Okay, suggest a reason why only 3 spots are detected on the final chromatogram. We can see this thing that there are 4 amino acids. They have mentioned this thing earlier. So why it is converting into 3? Okay, why we can observe only 3 spots? The answer is that as these 3 spots are there, it is telling you that 2 of the amino acids have the same distance traveled. So 2 of the amino acids are overlapping each other. So basically there are 4, but 2 have traveled the same distance. That's why they are overlapping each other. Okay. Next part of the same question. That's why I have repeated this question, as I couldn't do this part in the previous video. The RF value of some amino acids are shown over there. So state how an RF value is calculated. Back to this. So for example, we have to calculate the RF value of this spot. Okay. So we have selected the center of the spot. We have measured the distance from the start line till the center, and we have measured the distance in the centimeter with the help of a ruler. And then we have measured this distance which is traveled by the solvent. Again, the same line will be selected as a start line, and then we have selected the distances from the start line to the distances covered by these. So one distance is the distance covered by the spot, whereas the other distance is the distance covered by the solvent, which is called as solvent front. So it is, for example, we are saying that this is a point A. So we will say that RF value of A is equal to distance traveled by A divided by distance traveled by solvent. So it is a smaller distance divided by larger distance. So smaller distance divided by larger distance. Okay. So in this way, we are going to calculate the RF value. So sometimes examiner is asking what is RF value. Let me inform you that as well. RF stands for retention factor. Okay, so how to calculate RF value, that I have told you. What is RF value? It is basically a ratio of the distance traveled by the spot to the distance traveled by the solvent. So this is the ratio of the two distances on the chromatogram. As it is a ratio of the two distances, so centimeter will cancel centimeter from the top and from the bottom, and as a result, there will be no unit for the RF value. Okay, state which one of the amino acid listed in the table is present on the chromatogram. Your answer should include the measurements from the diagram and the calculation to justify your decision. Okay, I am back on this page right now. We can measure the distances. We can measure the distances with the help of a ruler. We have to calculate the answers for all of them. Okay, so for example, distance traveled by A divided by distance traveled by solvent. Here is distance traveled by B. Look at this. We are considering it as B. Distance traveled by B divided by, I am cancelling this line because this is not straight. Okay, this is again not straight. Oh, some issue is there. Okay, yes, this is right. Okay, so this is the distance traveled by B divided by same solvent. Okay, and now I'm taking the green color to make a differentiate between all of them. Okay, so this is called C. So distance traveled by A divided by distance by solvent, distance traveled by B divided by distance by solvent, distance traveled by C divided by distance by solvent. Hence, in this way, you will be able to find out three different types of RF values, and then you are going to compare it with these values. Okay, and then you will say this thing that this is present over there, either it is A, B, or C, but this is present over there in that chromatogram. That's why we are able to say this thing. And how we are able to say this thing? By using these measurements. Okay, then there is again another past paper question. Name the processes used to separate ethanol and mixture from ethanol and water. Okay, so it is called as fractional distillation. Fractional distillation. Why? I have told you this thing, distillation. Okay, distillation. I have told you this thing that it is a fractional distillation which is based upon the physical property of boiling point, and ethanol has a boiling point of 78 degrees Celsius, whereas water has a boiling point of 100 degrees Celsius, depending upon the physical property of boiling point. And the fractional distillation is basically dealing with those separations in which the boiling points are very close values. They have a close value. So here 78 degrees Celsius and 100 degrees Celsius are not very far apart from each other, but there is one more point, that ethanol and water are 100% miscible into each other. It means they are soluble into each other. So ethanol will boil off at 78 degrees Celsius when temperature will be above 78 degrees Celsius. Then the remaining in the mixture flask will be water only, and ethanol will be collected into the receiver flask. So they are saying why the process is suitable. Okay, name the process and why the process is suitable. So you have said this thing, fractional distillation, and then you have to explain it why as well. That I have told you, I'm telling it to you again, that basically fractional distillation is used to separate those two liquids or gases which are 100% soluble into each other, and they have close boiling point values. Same is happening over here. They are close boiling point values. They have close boiling point values. And secondly, they are 100% soluble into each other. So that's why fractional distillation is used for this purpose. Then describe a suitable method. Describe a suitable method in each case to separate the name Named substances from the mixture. Explain your choice in each case. Pure, dry, pure dry sodium chloride from a mixture of sodium chloride and sand. We have studied this topic in the separation techniques where we have taken this whole mixture. We are passing it through a filter paper, and before that we have taken this whole mixture. We have added water into it, and what will happen? Water will dissolve sodium chloride, and sodium chloride will become part of water. It is making sodium chloride solution, and sand will be settled at the bottom. Now what will happen? We will do the filtration. Sand will be collected at the top, whereas sodium chloride solution will be at the bottom. Now we are going to, after filtration, we are going to shift sodium chloride solution from the beaker to the china dish, and here we are going to heat it. This is tripod stand and this is burner, Bunsen burner. Here it is sodium chloride solution, and as a result of this heating, what will happen? All the water will be evaporated and only sodium chloride will be left at the bottom. Okay, they are saying pure dry, so you cannot prepare sodium chloride solution and can say that it is separated from sand. No, we have to take sodium chloride from sodium chloride solution as well. We have to take it out. Okay, now they are saying that a food coloring from a mixture of three food colorings. Okay, food colorings can be separated from each other with the help of a chromatography experiment. We will take this chromatogram, and as we know this thing that food coloring will be soluble into water. So what we are going to do here, I am going to make water, for example. First of all, okay, this is water, and this is the start line on which we have just placed food coloring. Food coloring will make different spots here, okay, mixture of three food coloring. So it means that we have to place three spots, and then there is again a spot, the line, it is the line which is traveled by, I should make a line here. Oh, I should make this paper a little bit bigger. Okay, this is it. Okay, great. So this blue line at the top is basically the solvent front, and this is called chromatogram. Okay, and this is the container in which this experiment is happening. As a result, three spots are developed over there. So then we can say this thing that three spots are obtained on the chromatogram. And do I need to draw the diagram? There is no need to draw the diagram, but a good student can draw a small diagram at the side of this. Okay. For example, this much space can be used for the diagram, and then you can write the details in this. Okay. This diagram with the proper labeling will improve the impression. Okay, so but this is not compulsory. Then you can say that you have to write the complete detail. A spot of the food coloring is placed on the start line, and the paper is immersed in the solvent called water, and then with the passage of time, water will travel up, and all the three spots of this food coloring, this three food colorings will be separated, and as a result, all three have traveled different distance, so these three are separated on the paper chromatogram. RF values should not be mentioned over here because they are not asking you to check the purity of the things. Okay, so in this way we can separate the three food colorings by using paper chromatography. Okay, thank you very much, guys. See you in the next class with the next session of the past papers. All the best to all of you. Allah Hafiz.